temperature gradient. Since such sampler requires careful adjustment and the air sampling rate is quite low, they are not commonly used. This type of samplers are not commonly used. Now we will discuss about the control of microorganisms in the air. How to control the microorganisms in the air. For that purpose, we require some methods or sanitation methods, air sanitation methods to be discussed. Methods for the continuous disinfection of air of enclosed surface have been employed and found to be practicable. These may be classified as separation of dust particles, particularly through the application of oil immersion to floor, blankets, and bed linens. Inactivation of organisms by mists or vapor sprays. Inactivation of organisms by ultraviolet rays. These are some of the methods employed for control of microorganisms in air. We'll discuss these in detail. That is separation of dust, or we can say physical. This is the physical method, and air contains different type of saprophytic as well as pathogenic microorganisms. Spores and vegetative cells of bacteria, fungi, and viruses particles are present in the air as free or in the form of droplets or droplet nuclei. Abiotic pollutants such as the dust particles, gases, smokes, etc. are also present in the air. It is essential to control microorganisms in the air and make it at least free of infectious microorganisms. Removal or killing of microorganisms in air by specific physical or chemical methods is called air sanitation or control of microorganisms. The physical method of air sanitation is use of UV radiation or ultra filters along with routine or domestic methods are some of the common physical methods. Of these, the dust control or separation of dust is one. The removal of dust in the room, cabins or hall is basically essential to control microorganisms in air. Dust found in homes, office, school, factories or hospitals arise from the airborne sand, ash or soot, soil and lint from bedding, clothing and carpets. Most dust particles are laden with a variety of microorganisms and have been studied particularly in relation to infections of respiratory tract and skin and secondary infections of burns and wound suppressions of dust wound. The separation of dust in room cleaning operation is therefore extremely important. Dust found in homes, office, the usual material dumped in room, unusable material dumped in room usually adheres with microorganisms, particularly the viruses. Such material should be possibly removed out. The dust along with the microorganism is removed by regular practice like sweeping with the brooms or vacuum pumps. Microorganisms on surface of floor, walls and roofs are removed or killed by washing with water followed by using detergent like Lysol, phenol or phenolic compounds. Proper ventilation of room is also essential to decrease indoor microflora of air. The next is use of the mists and sprays or chemical for the killing of microorganisms in the air. As such, the chemicals in liquid form are difficult to apply on walls, roofs, and internal parts of the machinery. Hence, the chemical compounds which appear in gaseous or vapor forms are of particular at particular condition are of significant for this purpose. Vapors of formaldehyde, ethylene oxide, and beta propanolactone are used for disinfection and sterilization of air in enclosed areas such as aseptic filling rooms of pharmaceutical and other industries, operation theaters, laboratories, and inoculation chambers. Hypochlorous acid and hypochlorite in a final concentration of 1:2 million is sufficient to produce 90 to 99% reduction in influenza virus suspended in air. In general, the same concentration applies to bacteria. The effectiveness of hypochlorous acid and hypochlorite aerosols from airborne organisms depend upon the moisture content. The next is formaldehyde, use of formaldehyde or gaseous formaldehyde. Formaldehyde is the simplest compound in aldehyde series. The characteristic feature of this compound is that at room temperature it polymerizes and exists in the solid substances, whereas at elevated temperature it depolymerizes to gas. The important polymer of is paraformaldehyde, a colorless substance which rapidly yields formaldehyde upon heating. It also marketed in aqueous solution as formalin, which contains 37 to 40% formaldehyde. Humidity and temperature have a pronounced effect on the microbicidal action of formaldehyde. In order to sterilize enclosed rooms or cabins, optimum temperature of 22 degrees Celsius and relative humidity between 60 to 80% is good. Formaldehyde, both in gaseous state and in solution, have high antimicrobial activity which kills all type of microorganisms. Vegetative cells are also killed more quickly than the spores. It is extremely reactive chemical which combines rapidly with vital organic nitrogen compounds such as proteins and nucleic acids and inactivates them. However, the fumes of formaldehyde are noxious. They are irritating to tissues and eyes. The vapors of formaldehyde have limited ability to penetrate covered surface. The next is beta propiolactone. Beta propiolactone in the gaseous form or effective use of mist and spray is good gaseous sterilizing agent with a wide spectrum of antimicrobial activity. It is sporadical, fungicidal, and virucidal. This compound is colorless liquid at room temperature with high boiling point at 155 degrees Celsius. Beta propiolactone is highly effective against microorganisms. Only 2 to 5 mg per liter of beta propiolactone is required to kill all type of microorganism. It acts on microorganism by inactivation of proteins and other biomolecules. It is non-flammable. However, it is vescient and lacrimotor, hence must be handled with care. It lacks the penetration power and appears carcinogenic. These are the drawbacks, restricts its use in the practical application. The next is ethylene oxide as a gaseous mist or spray. Ethylene oxide is one of the best chemical antimicrobial agent used to control microorganisms in air. It is a simple organic compound characterized to exist in liquid state below 10.8 degrees Celsius. It vaporizes rapidly above this temperature, hence it can be applied in its gaseous form at normal room temperature of 25 to 40 degrees Celsius. Vapors of this compound in air are highly inflammable. To avoid the danger, it is generally mixed with carbon dioxide or ferrons, which serve as an inert diluent and decreases inflammability of the ethylene oxide. Ethylene oxide causes alkylation of organic biomolecules such as enzymes, other proteins containing sulfhydryl group. Ethylene oxide is a unique and powerful sterilizing agent. The usual concentration of ethylene oxide for sterilization purpose is 400 to 800 mg per liter. It is very effective in destroying microorganisms on surface. It has remarkable penetration power and hence it is suitable to sterilize large packages of material, bundles of cloth, and even plastic. It has a broad spectrum of activity against microorganisms, including spores of bacteria and fungi. The next is a laminar airflow system or high efficiency particulate air filter system, that is HEPA filters. The high efficiency particulate air filter or HEPA filters are widely used to remove microorganisms In air, HEPA filters are composed of a mat of randomly arranged fibers composed of fiberglass filters with pore size of 0.5 to 2 microns. These filters provide filtration of smaller and particles and microbes than the traditional vacuum cleaner filters. Laminar airflow system generally involves the use of such filters. Air is blown through these filters and microorganism-free air is obtained. It is significant for inoculation of microbial cultures for different purposes. These laminar airflow systems are used extensively in microbiology laboratories, other research laboratories, hospitals, and aseptic filling rooms in pharmaceutical industry. Horizontal and vertical type of airflow models are available in the laminar airflow system. The laminar airflow cabin is first disinfected with suitable disinfectant like Dettol solution. Sterilized airflow and UV tube is made on for 5 minutes before starting the inoculation procedure. Care must be taken to strictly avoid the contact of UV with hands and other body parts. The UV tube is made off before opening the glass door. Fortunately, the UV lamp cannot pass through the glass of the cabin due to its shorter wavelength than that of the visible light. Gas burners is also provided in the cabin to sterilize nichrome wire, glass rods, glass spreaders, forceps, etc. and for cleaning the neck of the conical flask or test tube in the laminar airflow system. In this system, air passes through the high efficiency particulate air filters, those consisting of cellulose acetate filter medium pleated around aluminum foil in the HEPA filters. Particles of as small as 0.3 microns are removed by these filters. The air is passed through the bank of these filters and into the enclosed cabin, so the entire body of air moves in the uniform velocity along with the parallel flow lines. The use of next is to control the microorganisms in air. The next is the effective use of UV radiations. The UV radiations are the invisible radiations of sunlight. They are generally filtered by atmospheric constituents such as ozone, clouds, and smoke. The UV radiations include all radiations with wavelength of 150 to 3900 Armstrong. Radiations with the wavelengths around 2650 Armstrong have been highest bactericidal efficiency. Many germicidal lamps are available which emits UV lights within the most effective range of 2600 to 2700 Armstrong units. UV lights are widely used to sterilize the hospital operating rooms, aseptic filling rooms, and of industries and dairies, pathology laboratories, inoculation rooms, and laminar airflow systems. The mode of action of UV lights is UV radiation are highly lethal for microorganisms. Among different biomolecules, the nucleic acid are most sensitive to these radiations and undergo denaturation. The common type of effect on DNA are single-stranded break and double-stranded break. The damage to nitrogen bases and dimer formation, that is thymine dimer formation. Since the nucleic acid are one of the most important biomolecules in the cell, their destruction results in the stop of the growth and death of the microorganism. The UV radiations centering around 2537 Armstrong units emitted by special lamp exert a pronounced destructive action on the bacteria, viruses, and other microorganisms suspended in air are killed by this radiation. However, they are highly germicide and they have a highly germicidal range in between 2000 to 2967 Armstrong unit with a maximum of 2650 Armstrong unit. Germicidal lamps with other on the other hand have available at all times for the production of rays at their predominant wavelength of 2500 37 Armstrong units, which is essential. So this is the topic, that is unit 1 here, microbiology, for the paper Applied Microbiology, BSc second year. Thank you for listening carefully. Thank you very much, and hope you understand this topic very efficiently. Please